So hello everyone, today we are going to discuss the glycolysis. Okay, so if you see in the respiration process, okay, in respiration process what happen here, oxidation of glucose happens. Okay, so first of all, glucose is break down to which pyruvic acid. Okay, and pyruvic acid is further break down to lactic acid. Okay, so glucose to pyruvic acid formation takes place through glycolysis, while pyruvic acid to lactic acid conversion takes place through Krebs cycle. Okay, and lactic acid is further reaction is proceeded through ET pathway. Okay, so first of all you should be know about what is glycolysis. So it is the conversion of glucose to pyruvic acid. Okay, this process is called glycolysis. Okay, and if you see the glycolysis, glyco means here glyco means here it represent that glucose. Okay, lysis means breakdown. Okay, lysis is breakdown. So simple, in this process hydrolysis of glucose takes place into pyruvic acid. Okay, so if definition is asked that what is glycolysis, so simply you can write down that breakdown of glucose to pyruvic acid is called glycolysis. Okay, so it is the simple. Okay, so it takes place in the cell cytoplasm. Okay, cell cytoplasm, and there is a no need of oxygen, so this process is called anaerobic. Okay, in which oxygen does not takes place. Okay, oxygen deficiency is called anaerobic. Okay, so in anaerobic condition this process takes place. Okay, so in this one glucose is break down to pyruvic acid, and number of one glucose converted to two pyruvic acid molecule. Okay, and okay, one term is called phosphorylation. Okay, so we will talk about when we will describe this. Okay, so if you see in this process what happen here, first of all glucose is converted to glucose 6 phosphate. Okay, glucose structure you should be know about that. It is the simple glucose molecule. Okay, simple. I have discussed the carbohydrate, so you should be first watch the carbohydrate. So on that lecture you should be know about the glucose structure. Okay, so it is the simple glucose structure, and in this, and one thing is that if you see in this, 1 minute now. Okay, if you see in this, when glucose, when any process in which breakdown of ATP to ADP, this is triphosphate, this is diphosphate. Okay, triphosphate means 3 phosphate group present in this ATP, while ADP 2 phosphate group is present. So means 1 phosphate group is released here. Okay, and this phosphate group is attached from this glucose, and which position 6. So if you see this glucose 1, if you numbering do, then 1 carbon this one, because this is the functional group here, 1 2 3 4 5 6. Okay, so in this phosphate group will hydrogen will be replaced here, phosphate will be joined here. Okay, so structure should be this one. Okay, so this is the, and in when phosphate group is attached from any molecule, then enzyme which takes place here, the end means the back side will be kinase. Okay, so in this hexokinase, because hexo means this glucose sugar is 6 carbon, so it is called hexokinase. Okay, so glucose to conversion, glucose when convert to glucose 6 phosphate, glucose is the structure of when H will be here, then this molecule is called glucose, and glucose 6 phosphate means 6 position hydrogen will be replaced by phosphate group. This is the phosphate. Okay, so this is the glucose 6 phosphate. Okay, okay, and in this the next process takes place. So in this, one phosphate group is attached from glucose molecule, it is further converted to fructose 6 phosphate. So fructose, structure of fructose is the, this is the aldose sugar, this is the aldose sugar, this is the ketose sugar. Aldo, this is the keto group, this is the aldehyde group. Okay, so this is called aldose, this is called keto. In this fructose 6-phosphate means this molecule converted to this molecule fructose 6-phosphate. So fructose, this is the fructose, and if you see this, then 6 carbon, where carbon is 6, this one. Okay, so in this phosphate group will attached here. Okay, so this is the fructose 6-phosphate, 1 2 3 4 5 6. Okay, so this is the means chemical modification takes place. This is the isomer. Glucose and fructose, glucose and fructose both are isomer. Okay, so here glucose changes to fructose, so this is called isomerase. Okay, because it is the isomer and it is converted to one to another. Okay, glucose to fructose, so it is called isomerase. Okay, so there is no need to must remind the enzyme, because enzyme here which is mentioned here is named according to the concepts. Okay, so when fructose 6-phosphate is converted to fructose 1,6-diphosphate, fructose 1,6-diphosphate means one phosphate will be attached to here, one phosphate attached to here. Okay, so fructokinase, because 2-phosphate is attached. I have talked earlier that when any phosphate group is attached, the last enzyme which involved here, the last word will be kinase. So fructokinase, since in fructose molecule phosphate group is attached, so it is called fructokinase. Okay, so that's why it is called. Okay, in this fructose 1,6-diphosphate is further converted to dihydroxyacetone phosphate and glyceraldehyde. Okay, fructose 1,6-diphosphate, fructose 1,6-diphosphate, so it is the structure of fructose 1,6-diphosphate. If you did, okay, this converted to this one. Okay, this is the glyceraldehyde is 3 carbon sugar, so this is the simple structure of glyceraldehyde, and glyceraldehyde 3-phosphate, so this is the glyceraldehyde 3-phosphate. Okay, and dihydroxyacetone phosphate, dihydroxyacetone phosphate. Okay, dihydroxyacetone phosphate, so you can write down one phosphate is here, so it is the structure of DHAP. This is the structure of glyceraldehyde phosphate, glyceraldehyde, this one, the glyceraldehyde 3-phosphate. Okay, so simply again what happened here, DHAP and glyceraldehyde 3-phosphate, since the reaction is reversible. Okay, so here isomerase enzyme takes place. Isomerase means this sugar is 3-phosphate, means conversion of this, both are isomer. Okay, so that's why isomerase enzyme involved here. Okay, so this process, in all these process, phosphate group is attached, but in this phosphate group is released out, so that's why ATP formation takes place in this process. While in this process ATP involved, okay, ATP consume here, in this ATP formation takes place. Okay, so you can write down the ATP use in this process, but in this process ATP formation takes place. In this process glyceraldehyde 3-phosphate convert to 1,3-biphosphoglycerate, 1,3-biphosphoglycerate. Okay, so 1,3-biphospho 2-phospho, if 1 3 1, this 1 2 3, so 1,3-diphospho 1,3-diphosphoglycerate, so its structure is this one, and in this process 2-NADP convert to 2-NADPH, 1 NADP molecule release 3 ATP molecule. Okay, since here 2 NADP involved converted to 2 NADPS, so that's why 6 ATP formation takes place. Okay, so 1,3-biphospho, in this 1,3-biphosphoglycerate further converted to 3-phosphoglycerate. Okay, in this 2 ATP converted further to 2 ATP. Okay, so here ATP formation, 6 ATP formation takes place between these steps. 2 ATP formation takes place in this step. 3-phosphoglycerate further converted to phosphoglycerate with the help of mutase. Mutase means 3-phosphoglycerate means 3 position, the carbon means 3, 1 3 position phosphoglycerate converted to 2 phospho, means 2 position. It is isomerized and attached to another 2 carbon. Okay, so that's why mutase enzyme takes, means mutation takes place from 3 to 2 phosphoglycerate. Okay, again what happen here, 2 phosphoglycerate converted to phosphoenol pyruvate and enzyme takes place, enolase. This is enolase. Okay, and phosphoenol pyruvate converted to pyruvic acid, and in this 2 ATP converted to 2 ATP molecule. Okay, so it is the simple, means it is the conceptual reaction. If you see, if you should be consider the, if you consider the chemical structure and what takes place, then you will be learn very easily this glycolysis process. Okay, so first you should be know about the structure. Okay, what changes takes place in the structure, where phosphate group is attached, from where phosphate group is released out. Okay, so you should be keep in mind. Okay, so in this bioenergetics means, this bioenergetics means how much ATP molecule consume and how much ATP molecule form. Okay, so total net of ATP molecule. Okay, so if you see in the step first, in this step first, 1 ATP molecule involved, means okay, converted to ADP, means here it loses one ATP molecule. Okay, so I have lost -1 in the first step. Okay, so first step 1 lose, second step in the second step it will takes place. If you see 1,6 diphosphate, means here I have not mentioned here, 1 ATP converted to 1 ATP, means 1 molecule consume here ATP. Okay, and fourth step this step, 2 NADP converted to 2 NADPs. I have also asked that 1 NADP released 3 ATP molecules. So here, since 2 NADP is converted to NADP, so how much molecule, since 1 NADP converted to 3 ATP, so 2 NADP converted to 6 ATP, so I have attached to 6 ATP. Fifth step, this one here, 2 NADP converted to ATP, so I have attached 2 ATP formation takes place, and the last step I have. Okay, so total involved ATP how much, total में क्या करोगे, you will consider both lose and gain. So 6 to 8 to 10 to 12, so total involved ATP 12, net gain कितना हुआ, 6 to 8 to 10 molecule ATP formation takes place. Okay, and total gain कितना हुआ, 10 minus 2 to 8 ATP formation takes place in the pyruvic acid. Okay, so question will be arises from this one. In the NEET exam, question arises from this part, how much ATP involved in the glycolysis. So answer will be 12. How much gain ATP from glycolysis, then 10. Okay, how much total form ATP, then 8. Okay, so it is the simple glycolysis process. Okay, so if you want to learn in easy way, first of all you should practice to draw the chemical structure of glucose, fructose. Okay, glyceraldehyde, dihydroxyacetone. Okay, after that you will consider from where you will be numbering of carbon. Okay, so numbering of carbon takes place according to priority of functional group. Okay, so if you have capable to draw the structure, then numbering is carried out by you, and after that enzyme, I have mentioned that kinase involved in when 8 phosphate group is attached, then kinase involved. So glucose is 6 carbon, so hexokinase is involved, isomerase is involved, kinase involved. Okay, so since in this phosphate group is attached, so that's why enzyme named as fructokinase. Fructo means fructose में जुड़ने जा रहा है तो fructokinase, glucose में जुड़ने जा रहा है तो hexokinase या glucokinase you can say. Okay, and after that dehydrogenase enzyme is involved where, when ATP released. Okay, when ATP formation takes place in the step, then enzyme which is involved here is named as dehydrogenase. Okay, so it is the simple concept of glycolysis. Okay, so I think you will be learn this glycolysis. Okay, So thank you for watching this video, and if you like this video, please subscribe my channel, because if you like this video, I will motivate it for further uploading next grep cycle. Or you can also ask to me which video will be uploaded again. Okay, so thank you. And if you have any queries from this topic, then please write down your comment in the comment below comment box. We will solve your problem. Okay, so thank you.